Hi, everyone. Um, Today we're going to shift gears. We have been talking about B cells for the last couple of lectures. B cells as the first example of a small lymphocyte that's vital for our adaptive immune response. Remember, it's because B cells make immunoglobulins. And at first, these immunoglobulins are expressed as surface receptors that stay anchored to the surface of a B cell. And then if it turns out that the immunoglobulin proves to be useful, meaning that the variable domains of the immunoglobulin molecule actually end up being the right shape to bind something, that something being some component of a foreign pathogen that's infecting our tissues, then that B cell may differentiate and change into a plasma cell that can then make more of that immunoglobulin, but importantly, make the form of the immunoglobulin that can float freely away from the B cell, and that free floating form is the antibodies. So the last couple lectures, we've been talking about how it is that these B cells can come up with different shaped immunoglobulins. When I say different shaped immunoglobulins, I mean the actual immunoglobulins are still pretty rigidly confined to being a standard Y-shaped molecule. But we said the important thing is you can vary the actual ends, the variable domains of those immunoglobulins through the process of VDJ recombination to come up with hundreds of millions of different variations in the hopes that some of them will be the right shape to bind a brand new pathogen. So we are now going to take a look at the B cell's cousin, the T cell. And we're going to take a look at the role that T cells play and how, in some ways, T cells and B cells are very similar, but they have very importantly distinct functions as part of our immune system. So a good way to go back and get us all on the same page is to look at this cartoon again that we always come back to and remind ourselves that B cells and T cells are closely related. They are certainly representative of our adaptive immune system, and they come from the same common lymphoid precursor, as do natural killer cells. So here's the B cell that makes its immunoglobulin, the Y-shaped antigen binding specific molecule that then, if it turns out to be chosen, can be made into free floating antibody forms when the B cell turns into a plasma cell. And remember that the way the B cell is able to make free floating antibody compared to the membrane bound immunoglobulins is simply through a process of different RNA editing of the RNA that gets transcribed once the B cell through VDJ recombination has put together different fragments to make one usable immunoglobulin chain gene. So for example, heavy chain or light chain. Right? Then when it transcribes the heavy chain, the end of the heavy chain has the information for either having the end that's going to anchor it to the surface or the end that's going to allow it to be free floating. And so the B cell can then simply do alternative RNA editing to either have the resulting molecule be stuck to the surface as a Y or floating away as an antibody, as a Y-shaped antibody molecule. So T cells, if you look at this cartoon, right, you can tell on the surface, it also has some kind of receptor, but right away you can tell, just the way the cartoon shows it, that it is different than your typical Y-shaped immunoglobulin. And if you notice, an important feature is this T cell, even after it sort of matures and becomes chosen, right, its receptor never leaves the surface, right? So you never have free floating versions of this receptor. So let's take a look and see what's going on there. So the receptors that we're talking about are what are called T cell receptors, right? Just like the immunoglobulins on the surface of a B cell are called B cell receptors. But T cell receptors, or TCR, I think that comes up a lot more in discussions about immunology You don't really talk about B cell receptors that much, but you talk a lot about T cell receptors and T cells. So hopefully many of you are already kind of intrigued about what T cells do. So to start off with, the T cell receptor, so let's start with the middle sort of panel of this cartoon here in the green square. Here's a T cell receptor, and a T cell receptor is also made of a variable region and a constant region, just like immunoglobulins. And in fact, the variable region, right, it's just like the variable region of an immunoglobulin. It's made of two chains together, but instead of being a heavy chain and a light chain, it's just two similarly sized chains that we just call alpha and beta. In fact, if you look at this cartoon and compare it on the left, so on the left is what we've been talking about the last couple lectures in immunoglobulin, right, the Y-shaped immunoglobulin, you realize, yeah, the T cell receptor kind of just looks like one arm of the immunoglobulin, right, just stuck directly to the surface. In many ways, that's what it is, right? I mean, that's how it functions. It functions like just being one arm of it. And you can see it's much simpler than the immunoglobulin. Right? Later on, when we talk about the evolution of our immune system and how these different things came about, hopefully you'll realize, right, that the simpler T cell receptor actually came first in evolution. Right? That makes sense. This is a simpler structure. Mother Nature, our immune systems evolved to make this first, and then later on, this idea of a antigen binding variable molecule, right, got improved upon and became the immunoglobulins or the antibodies. So B cells came later on in evolution compared to T cells. So you have a variable region, a constant region, and then you have this transmembrane region that anchors it and keeps it anchored to the surface of the T cell, right? And just like the IgGs, it's the combination of two different surfaces, one from the A chain and one from the alpha chain and one from the beta chain, that combine to make the three-dimensional shape that will recognize some sort of target molecule. The other distinction is, so number one, I said that there is no soluble free-floating form of T cell receptor, right? So that's very important because that's very different than antibodies that are made by B cells. And the important thing is that T cells do not undergo any kind of hypermutation to try to improve the fit of these or the binding affinity of its variable domain. Right, we spent quite a bit of time last time looking at how B cells, once they're selected, right, are encouraged to increase the binding affinity of these through processes of random mutations, right, which is pretty amazing. But T cells don't do that. So whatever T cell receptor T cell has, that's all it has. Okay. On the right is a more realistic ribbon diagram model of a T cell receptor. So the purple one on the left is the alpha chain. So here's the variable region of the alpha, the constant region of the alpha. And on the right, the blue one is the beta chain. Right? So you have the variable and the constant. And then up here, you can see, right, it's the contribution of these three loops from each chain that makes the unique three-dimensional surface on top here that some molecule will hopefully interact with. Right? And this T cell that carries this receptor will then recognize the presence of that molecule. Okay. So not surprisingly, T cells make their T cell receptors and their variable domains through the VDJ recombination process. I already told you, T cells actually are earlier in evolutionary history than B cells. So in reality, right, this is the first time in evolution that any of our cells did VDJ recombination. So keep in mind, though, that these are different loci or different gene fragments than the ones in our immunoglobulin gene loci. It's a little confusing because the alpha chain of the T cells, the group of little fragments where you find VDJ happening to make the alpha chain is also on chromosome 14, right? But here you see there are way more variable blocks. Remember, in the B cells immunoglobulin loci, there was maybe like 30 red blocks to choose from, right? And maybe like 20 or so yellow J blocks to choose from. Here you can see in the T cell receptor or T cell alpha chain locus, you have like between 70 and 80 of these red blocks and like 61 of these yellow blocks to choose from. That's a lot for the big oven mitt to actually choose from, right? So you're going to get a lot more variability just by your choices of the blocks. So an alpha chain is a V block, a J block, and then a C block. So here in the middle, right, this cartoon is kind of drawn so that you go from the top towards the middle and the bottom towards the middle. The alpha chain then is made this way. The beta chain is kind of like the heavy chain locus in B cells in that it now has this extra green D block, right? But again, much more variability. 52 of these red blocks to choose from, right? And then you have an assortment of J blocks and D blocks, and that makes the beta chain, okay? So the T cell receptor then, when it's made and it's completed, and it's on the surface of the T cell, it would be like this. So the blue chains here are the alpha and beta that were made by VDJ recombination, and now the T cell is making this central T cell receptor. But these two chains don't function by themselves. One important thing to keep in mind is that this part that sticks into the membrane that anchors the alpha and beta chain to the membrane, it's pretty wimpy, right? Because look at how little of it is represented as being actually sticking into the cytoplasm. Right? For any cell surface receptor to work, the principle behind it is on the outside, something is going to bind to this pair of chains of polypeptides, right, proteins, and that's going to cause changes in the protein that are reflected in the part of the protein that sticks into the cytoplasm, also changing its shape. And that's how it then begins to communicate or interact with other proteins in the cytoplasm. Right? So if you just have this wimpy little bit of the protein, that navy blue thing sticking to the cytoplasm, that's not a lot of surface to have a lot of interactions with it. Therefore, a functional T cell receptor is associated with these other proteins, these yellow, orange, and red proteins that are known as the CD3 complexes. So each of these have an epsilon, a delta. On this side, you see a gamma and an epsilon. And what they do is they stick to the T cell receptor, and their cytoplasmic domains, right, the ends that stick into the cytoplasm, are much bigger. In addition, you have one that's known as the zeta chain, these green ones, that are almost like all about just being a major cytoplasmic portion of the protein that also associates. And together you have a complete T cell receptor complex. So why is this important? Because the much more pronounced cytoplasmic domains of these proteins provide the signaling. So when something binds to the top of the T cell receptor here, and the T cell receptor changes shape, imagine it as kind of like a ripple effect, right? The shape of the protein starts changing because suddenly you have something up against it, and the peptide chain, all the side chains of the amino acids, they have to kind of adjust a little bit to get comfortable again. So that kind of like trickles down in here. That causes these associated proteins like the CD3 proteins and the zeta chain proteins to also change their shape. And that's where the main cytoplasmic signaling happens. Okay. Just a quick story. You know, when I graduated from undergrad with my biology BA, I took a fellowship position at the National Institutes of Health out in Bethesda, Maryland, just like outside of DC. So that's the NIH, right? That's like the big mothership of research funding here in the US. And it was a great place. I loved the NIH. But anyhow, I got accepted into a lab there at the NIH that was part of the National Institute of Child Health and Human Development. And the lab that I was part of studied the development of the zeta chain as part of the immune system properly maturing and being able to do things like have good T cell activity. At the time I was 22 and I was just happy to be in a lab there. It was exciting being at the NIH in Washington, D.C. And I got to work with mice. And so the lab, we would generate these transgenic mice, meaning mice that were engineered to have alleles or particular genetic variations in their zeta chain genes that were meant to reflect certain genetic variations that were seen in people, right? And we wanted to understand what impact do these genetic variations have. And so we would breed these mice, and then I was in charge of kind of maintaining the mice. And I'd also take tissue samples from the mice to do southern blotting to identify which of the mice in the litter of mice that were breeding actually had the mutations, right? And then we'd study how these mice responded, how their immune systems developed, how they worked. Now, the funny thing is, at the time, like I said, I had not learned any immunology at all. I mean, you guys at this point in the semester know way more immunology than I did when I started in this fellowship position at NIH. But I learned how to do all this stuff, and and I learned, that was the start of me learning immunology. And so at that time, I would have never imagined that years later, my career would lead me as a cancer biologist to really understand, to be trained in immunology, and one day to be teaching immunology to students, right? So, so it's kind of funny. A lot of times, life goes in a full circle, and you never know where you end up. So I always think about that, right? Because as a 22-year-old, I was trying to figure out this whole zeta chain thing, but I was just having fun, right? Hanging out and breeding mice and stuff like that, and running gels. All right. So T cell receptors, then, just because they have way more V blocks and J blocks to choose from, right off the bat, you have a lot more variability just because of your choices of gene fragments to put together, compared to something like the immunoglobulins in B cells. This table is a little confusing, so I'm not going to go over all parts of it, because some of these are things that we won't talk about until later on, and even then, I'm not sure why the book kind of presents it this way. But there are a few things that I wanted to point out to help you compare and contrast. So the left part over here is talking about the immunoglobulins that B cells make. And then the right part talks about the alpha beta T cell receptors, right, that we are learning about in today's lecture. So remember, the immunoglobulins have a heavy chain, so that's the H, and they have a light chain, either a kappa or a lambda light chain. Right? And the alpha beta T cells, they just have a beta chain and an alpha chain. If you look at the number of V blocks, or the red blocks in the cartoons, right, to choose from, typically heavy chain, light chain immunoglobulins, there's like 40 or 70 to choose from. In alpha beta T cells, you see there's more variable domains to choose from, about the same number in alpha chain. Diversity segments, the D segments, in the immunoglobulins, you actually have higher number of the little D, the green blocks, compared to the beta chains in the T cell receptor. But really, if you go down to the J blocks, right, these are the joining segments here, you'll see there's way more of these, what would have been in the cartoon yellow blocks, in the T cell receptor gene loci, compared to the, I don't know, this is saying there's like, what is this, 15 or so together, different J blocks, okay? When you take that into consideration, right, and remember, it's still VDJ recombination, so you have the big oven mitt choosing a red, a yellow, a green block, putting it together, and then the loop of rope is cut, right, and then the ends of the rope, the DNA that's been cut, has to be joined together through random DNA bases that are being added, right? That part's the same, but because of the higher number of different blocks that you have to choose from, right, that really blows the probability number up very high. Okay, so junctional diversity, then, as a result of the VDJ recombination, in B cells, it's like around 3 to 10 to the 7 different variations of immunoglobulins you can get at that point, whereas in T cells, you're up to like 10 to the 11. Right? That means the total diversity, once you think about in immunoglobulins combining the number of possible heavy chains with the number of possible light chains, that's where we get our 10 to the 12 or 10 to the 13 variations, right? But in the alpha B cells, you get 10 to the 18 possible variations, right? So that's like 5 orders of magnitude larger. So just from the process of making an alpha beta T cell receptor, there are way more different shapes of these T cell receptors than there are immunoglobulins. So that's part of why T cells don't have to worry about hypermutating, okay, or why hypermutating to change these things around isn't as big of a deal. So this is a very important concept here. All right, so if I kind of lost you on the previous table, you kind of zoned out, pay attention to this, right? One of the important, very important distinctions between how a T cell receptor can detect a target versus an immunoglobulin in a B cell is that, remember, immunoglobulins, the Y-shaped immunoglobulins, they can bind anything just as long as that anything, the epitope on that anything molecule, is the right shape, the right three-dimensional shape that the ends of the immunoglobulin arms fit. Right? So that could be a protein, it could be a carbohydrate, it could be a lipid, right? T cells, on the other hand, are much more restricted in what they can bind. Right? They're limited to only binding short peptides. In other words, they're only able to bind small pieces of protein, right, as the thing that they try to identify, right? So that's very different than what a B cell does. Where do these peptides come from? These little fragments of proteins? Well, usually it's from, if we're talking about an immune response, we're talking about small pieces or fragments of proteins that came from a pathogen, right? Usually not a complete protein, but a protein that's somehow been processed and chopped up into small little pieces, short peptides. And the other key thing here, right, is that if you have a cell that has this foreign protein in it, that's processing it, right, and little pieces of the protein are coming off as little peptide fragments, then these little fragments actually have to be displayed on the outside of the cell in order for a T cell to come along and have its T cell receptor and have a chance of taking a look at the small peptide. So how do things get presented? That's what this cartoon is showing. Okay, we'll go into this in a lot more depth, but this is the first little intro cartoon here. Let's say this gray cell is a normal cell in your body, right? It's a perfectly normal cell, and somehow, right, there's a foreign protein in it. So this is a protein that maybe came from a pathogen, right? And so normally the protein is all folded up in its normal three-dimensional shape. But if this protein gets broken down, for example, if the cell itself is able to process this protein and chop it into small little pieces, right? These small little pieces would be peptide fragments of different lengths. Some of them are longer, like the hot dogs. Some of them are really short, like the red dots. Right? So it's the short little ones that get presented on the outside of the cell. How does the cell do that? It uses a special protein platform known as MHC, right? Or major histocompatibility complex. So MHC is a three-dimensional protein shape that serves as, I call it a platform because that's what it is. It's able to hold on to the red dot, and importantly, it's able to present that red dot, right, to the outside. The last panel here, you see a blue T cell that's passing by. And on the surface of the T cell is its T cell receptor, in this case, an alpha beta T cell receptor, let's say, right? And it is then the right shape in its variable domain to bind the red dot that's being presented to it on the yellow MHC by the target cell, right? So this big blue cell, well, not big blue, but this blue T cell is coming by. Here you have this gray cell that's presenting small little pieces of peptide on its surface. The T cell presses up against the gray cell so that the T cell receptor has a chance to check out any of these red dots that are being shown off on the surface of MHC. So here are three important terms. Antigen processing, right? That just means the breakdown of proteins, or in this case, the breakdown of proteins to be able to actually come up with small little pieces that can serve as identifiable targets. Antigen presentation then is transporting these little fragments from the cytoplasm of the cell to the cell surface, right, and presenting them on MHC. And MHC itself stands for Major Histocompatibility Complex. I know that's a real mouthful, right? And you're like, what a weird word. Why don't they just call it yellow platform? No, Major Histocompatibility Complex. It's a complex of proteins, right? And early researchers identified these based upon the fact that it seemed like these played a very important role in tissue rejection. In other words, when tissues are being transplanted from a donor to a patient, right? I think all of you are aware that there's some kind of matching that has to happen beforehand. Otherwise, the patient's body might reject the donated tissue. And so even though it might seem obvious, you don't think about, yeah, there was a time when people didn't know that, right? And didn't understand how that works. And so when they finally discovered that there's a group of genes that code for a group of proteins that are the major determining factors of histocompatibility, right, the compatibility of tissues, right, they named then these groups of protein complexes the MHCs. Right? So we'll look at that as we go on. We'll look at the genes themselves. Importantly, one thing I want you to notice is the way this cartoon is drawn. You can see the blue T cell receptor is not simply recognizing the red dot. What is it also recognizing? It's also recognizing the three-dimensional surface of the MHC, right? So that implies that the VDJ recombination process that this T cell went through to come up with its unique T cell receptor recipe, right? Part of that shape information is also recognizing the yellow MHC proteins that are being used to present the red dot by this gray cell. There are two classes of MHC. Right? Fortunately, for simplicity's sake, we just call them MHC type 1 and MHC type 2 in Roman numerals. MHC class 1, MHC class 2. Right? How a peptide, a red dot, is presented on a cell, either by MHC1 or MHC2, has profound implications on what happens next in terms of the type of T cell that will respond to this. Why? Let's think about this for a moment. If you have a normal, healthy cell in your body, like an epithelial cell from the lining of your respiratory tract, and it gets infected by something like a virus, right? That means the cells themselves are being taken over by the virus. Those virus particles will be in the cytoplasm of your cell. And that means all the little fragments of proteins that come from the virus, there'll be traces of them inside the cytoplasm floating around. Okay. Let's say a different scenario. Let's say you have a cell that's not infected by the virus, but let's say you have a cell that's going around and its job is to eat the virus. For example, a macrophage, right? Or a dendritic cell. In that case, those cells are not necessarily in jeopardy, right? Because they are not infected. They've just eaten a virus particle and have also taken apart or processed the antigens of the proteins. So those little fragments of proteins wouldn't just be floating around in the cytoplasm, right? They would already be in these special little vesicles. Right? So that's a big distinction. Depending upon where those fragments came from within the cell, either freely floating around in the cytoplasm, like a cell that's been infected, or from within special vesicles from a cell that has eaten, let's say, the virus, right? That will influence or determine which type of MHC platform, 1 or 2, ends up being the one to present the red dot. Okay. So if you see here, this is just obviously a very simple cartoon, but you can see the main structural differences between MHC class 1 and MHC class 2 is that MHC class 1 is made up of one long what do you call it, segment or peptide. Oh, not, I don't want to call it peptide because the red dot they're saying is a small peptide, but one long yellow chain that starts from the upper right here, continues through, these are 3 separate sort of domains of the protein, right? And then it ends up being the anchor and this extra globular protein that is just kind of stuck on there. Whereas MHC class 2 is made of 2 more similarly sized chains that are in parallel, and you don't have this extra separate globular protein stuck to it, right? So structurally then, MHC1 and 2 are similar in the sense that they both have this kind of cleft up at the top where they can hang on to and present the peptide, right? The foreign peptide, the small piece of a protein that came from the pathogen, but the other parts of it are a bit different, right? And that'll be very important for how T cells recognize what kind of MHC they're looking at, right? So the different MHC type tells the receiving T cell that's going to recognize this with its T cell receptor what to do with this red dot in terms of how to interpret what the T cell is encountering. Okay. Here are the structures again, just to kind of illustrate this a little better. In MHC class 1, again, you have one long alpha chain, this A chain that starts from up here, and it goes through these 3 globular domains, right? And then you have this separate beta microglobulin, beta 2 microglobulin that's stuck up against it. So here's the structure, meaning somewhere over here, you can find, this is probably the end terminus of this. It probably winds around and makes the alpha 1 part, then it keeps winding around, makes the alpha 2, and then it ends up down at the alpha 3, and somewhere down here is the very end of it, right? Probably tucked behind here, right? And then you have the separate green microglobulin stuck to it. Compared to MHC class 2, where both of the chains are more or less the same size, right? And they're just kind of intertwined. Notice at the top of both, you have these alpha helical regions that represent the cleft in which this thing can hold onto a small peptide that came from a pathogen. All nucleated cells in our body express MHC type 1, right? And that kind of makes sense if you think about the fact that all nucleated cells in our body, there is a chance that that cell could be infected by something, right, from the outside, like a pathogen. So therefore, all cells in our body, with the exception of certain cells like red blood cells, things like that, that don't really have a nucleus, right, have on their surface MHC1, ready to present the red dot if there's a red dot, coming from inside the cell. MHC2, though, is different. Only a small subset of your body's cells bother having MHC2 on their surface in addition to MHC1. This subset of cells, not surprisingly, are some of your immune cells. So we call these immune cells professional antigen presenting cells, right? Or APCs. These include things like macrophages, dendritic cells, and B cells. Right? In particular, macrophages and dendritic cells, these are cells that work by phagocytosing things, right? So already, hopefully, you can begin to see what's going on here, right? Cells that are immune cells that do their job by taking in pathogen to destroy them, right? Those cells want to also present what they've found on their surface so that other immune cells have an opportunity to learn from it. But they want to present the red dots on their surface in a different way than simply putting them on MHC1, because all cells in your body can present these red dots on MHC1, but that would mean that those cells are saying that they are infected by the pathogen, right? A macrophage, a dendritic cell, it does not want your immune system to somehow improperly think it's infected, right? Because it's just being like the messenger here, right? It found something and wants to pass this message on. It itself does not want to be targeted for destruction. So that's why this third bullet point here, I say, think about why this makes sense in the context of a peptide that came from intracellular infection, right, versus a peptide that got into a cell because the cell was a professional APC and ate it, right? The subsequent steps on what the immune system does with this are different depending upon those two scenarios. Here we are looking straight down at the surface of a cell at these MHC platforms sticking up at us. And what we're looking at is just part of it, the part that represents the cleft on the top that's going to hold onto and present the red dot, right? So here, of course, the red dot is a little bit more detailed because we already know it's not a red dot. It is a short sequence, a short peptide sequence, right, that's being shown off. So here it's a small chain. In MHC class 1, you typically find small peptides of about 8 to 10 amino acids long, right? So this would be like a 10 amino acid or something peptide chain. These little things sticking off of it are the little R groups of the different amino acids, right? Compare that with the MHC class 2 platforms. You're looking straight down at it. They can bind peptides that are a little bit longer, like 13 or more amino acids, right? That's why this is longer than the one up here. Notice also how in the MHC class 1, right, the structure is all green, because remember the MHC class 1 is built by one long chain that has this extra sort of thing stuck to it. So the top part, right, the actual cleft is all part of the same chain. So it's all in the green here, one same color. Whereas in the MHC class 2, it's two separate chains that contribute half of the cleft at the top. That's why one is yellow and one is light blue. Right. How then does a T cell that comes along and has a T cell receptor on it, right? How does it actually interpret the fact that this is MHC1 that's showing the red dot to the T cell versus MHC2, right? Because remember, that makes a big difference in terms of what the message is about what should happen next. So here you have an example of two populations of T cells. One group of T cells is what we call cytotoxic T cells. The other group is what we call helper T cells. And as the names imply, cytotoxic T cells, their job is to synapse or bind to a target cell, check out what's going on, right, by looking at what that target cell is showing it, and then potentially, if it doesn't like what it sees, right, or I should say if it likes what it sees, if its T cell receptor binds, then it may kill the infected cell. Or I should say, tell the infected cell to die, right? Because T cells, just like natural killer cells, right, they have the ability to release cytotoxic messages that stimulate the target cell to undergo apoptosis. So what we see here is that both the cytotoxic T cell, the purple cell, and the blue helper T cell, they have alpha beta T cell receptor on their surface. And it may actually be that by chance, the shape of these alpha beta chains might be identical in both, all right, just by chance. Okay. So it will then bind to, here's a target cell up here that's displaying the red dot, a short peptide, being carried on MHC class 1, right? You can tell, because remember, MHC class 1 is made up of one long chain and then this beta microglobulin stuck up against it. The cytotoxic T cells can tell that this is MHC1 because it has another type of co-receptor on its surface to help interpret what kind of MHC this is. And that receptor is called CD8. Right? So CD8 in purple here is also made up of two chains. Right? And these are the right shape so that when the T cell receptor binds to the MHC, the CD8, which is also on the surface here, it cruises around and it bumps into this. It's attracted to the T cell receptor, and it is the right shape to bind to the shape of the MHC1, right? And that combination of the T cell receptors, light blue alpha beta chains signaling through the CD3 and the zeta chains into the cell, plus the positive signal of the CD8 co-receptor also detecting that this is MHC1, will activate the cytotoxic T cell, and then that cytotoxic T cell will kill the target cell. Right? Again, because the red dot that came from, let's say, bacteria or virus or whatever, ended up being shown on MHC1 because this cell was infected by it. So the red dots were just floating around in the cytoplasm. To contrast, what if it's the exact same red dot, right? But it's coming from a professional antigen presenting cell that was not infected by the bacteria or virus, but it phagocytosed the bacteria or the virus, right? So this antigen presenting cell, it's still healthy. It's not in trouble. In fact, it's just doing its job really well, right? And it presents that red dot that did not come from floating around just in the cytoplasm, but the red dot came from inside of the special vesicles where the virus or the bacteria was digested inside of the antigen presenting cell. It presents the red dot on MHC class 2. So this T cell over here has a T cell receptor that by chance recognizes the combination of the red dot on MHC, right? But instead of CD8 on its surface, its co-receptor is CD4. And CD4 then swings over, and it's the right shape to be able to bind to and recognize that this is MHC2. And therefore, that combination of T cell receptor activating with CD4 activates this helper T cell. This helper T cell, right, just like the other T cell or just like natural killer cells, it's going to release cytokine messages, right, for this antigen presenting cell to receive. But these won't be the pro-apoptosis, you better hurry up and kill yourself messages. These will be cytokine messages that encourage the antigen presenting cell to do something more, right? For example, to increase its activity, or perhaps differentiate and do something else entirely. So we have these two main populations of adaptive T cells in our body, the CD8 positive, right, and the CD4 positive. Okay. Each one of these only has either CD8 or CD4 on their surface. You're not going to have one that has a mix of the two, because that's how these two types of T cells can tell what the message is that's coming from the cell that they've bumped up against. Right. Let's think about it this way. Right. Here we have, here's bad clown. And if bad clown somehow gets into one of the cells of your body, for example, if he infects one of your cells, then pieces of bad clown will be presented, as the red dot on MHC1. Right? If bad clown is phagocytosed and destroyed inside of something like a macrophage, that macrophage will present the red dot from bad clown on MHC2. Right? So you can think about it this way. The MHC1 versus MHC2 provides different information, right? And is going to tell whatever T cell comes and recognizes it, either the CD8 positive T cell or the CD4 T cell, right? How to interpret the presence of this red dot. It's like these driver's licenses here, right? So here's two Hawaii driver's licenses. One on the left is kind of your typical one. This is the one that, I think everybody has. This one over here, I'm not really sure what this is. It's called a limited purpose driver's license. I don't know if this is like a probational, like when you're learning how to drive. I didn't grow up here, so I don't recognize this, but maybe you guys do, but it's not acceptable for official federal purposes, right? So I guess you can't really use this as a official ID. The point is, the photo of bad clown, in other words, the identity is identical, being presented on either one of these MHCs, but the extra bit of information, in other words, which MHC it's on, is like the actual driver's license card, right? It contains additional information that influences how the card can be used. So a single T cell then, right, and this is crucial that I get this point across, is equipped to read only one kind of ID card due to that T cell either having, in addition to its T cell receptor, of course, CD4 or CD8 co-receptor on its surface to help out and bind to the MHC and actually interpret which MHC type, 1 or 2, the T cell is actually encountering. Right. And the reason I say that's important is because students often ask me, well, if you have an infected cell that's presenting this red dot on MHC1, and you have a helper T cell that has the right T cell receptor to recognize this red dot sitting on MHC, does that mean that the helper T cell won't actually recognize this? No, it will recognize it because its T cell receptor is the right shape. But once the CD4 kind of swings over, right, the CD4 co-receptor is not going to recognize MHC1. So as a result, that helper T cell just moves on and doesn't do anything. All right, because remember, these cells can all be interacting. It's just whether or not the right combination of signals takes place so that the next step can happen. So here's another illustration of this to help you visualize it. Right. So what we're basically saying is that if you look at a cytotoxic CD8 positive T cell versus a helper CD4 positive T cell, they may actually have T cell receptors, the light blue T cell receptors, that are more or less identical. In other words, they can recognize the same foreign peptide, right? And that's why in either of these cases, the red dot might be the exact same red dot in these different situations. Why would that happen? Well, because remember, in your body, there's all kinds of cells that are present, right? So some of them, if you have a virus infection going on, would be like the one on the left that is actually being infected, right? And it needs to raise the alarm that it's being infected, right? Because the cell is being taken over and it's going to start making more virus particles. So it's presenting the red dot for CD8 cytotoxic T cells to see, right? And then that would cause the CD8 cytotoxic T cell to kill this infected cell before the spread of the virus, gets out of control, right? Versus at the same time, in your body, you have macrophages and other things that are picking up on the virus through phagocytosis. So they are going to be communicating by presenting the red dot, right, on MHC2. So that helper T cells are the ones, if they pass by, have a chance to learn from this, right? And not only will the helper T cell give feedback to the professional antigen presenting cell here, but as we will learn, the helper T cell will also coordinate many other functions as well. So I'm going to stop here because we actually will continue with this discussion on Friday. So I'm going to save this slide for Friday's lecture. Okay, but this is just a summary of different scenarios that are meant to help you kind of understand this important distinction. So if you want to, you can take a look at this now and see if you can and figure out what's going on. But don't worry, we will start with this on Friday. Okay. All right. So let me wrap it up.